The first thing that we did in this lab, besides gathering up the materials, was to cut up the onion. And the reason we did this was to break apart the plant cell walls that the onion had, and to allow the extraction solution, which will be introduced later, to get inside the cell. The next thing that we did was to introduce the onion to a solution consisting of shampoo, salt, and baking soda. The shampoo in the solution broke apart the nuclear membrane, allowing the DNA to leave the nucleus, something it can't usually do. It also broke apart the cell membrane, allowing for DNA to leave the cell altogether. It did this by dissolving lipids and proteins, which made up both the nuclear and cell membranes. The salt in the solution neutralized the negative charges on the phosphate groups found on the backbone of DNA to the point where DNA is still dissolved in water, but was able to precipitate in the presence of alcohol. Salt also causes the lipids and proteins to precipitate without alcohol, which is important for straining purposes as they will mostly get strained out. The final component of extraction solution was baking soda, which was used to keep the pH of the solution around neutral, which was an environment that DNA is best suited for. The solution containing the onion and extraction solution looked mostly clear before shaking, and then after shaking it looked extremely frothy due to the shampoo bubbling. After straining, the solution looked very yellow. The next thing that we did was filter out all the unwanted excess material from the onion/extraction solution mixture. This unwanted material includes proteins and lipids from the cell and nuclear membrane, as well as various other parts that make up an onion cell besides DNA. Only liquid could pass through the filter, and since the proteins and lipids were in precipitate form, and the DNA was dissolved in the water, DNA was able to pass through while the other materials were filtered out. After filtering out all the excess material, we introduced isopropanol to the DNA, which caused it to precipitate. The introduction of salt started this precipitation process by making the sugar phosphate backbone of DNA less negatively charged, and therefore less hydrophilic. The isopropanol then further reduces DNA's hydrophilic nature until it drops out of solution. We saw the formation of a precipitate after infusing alcohol during the lab. The precipitate that we saw formed was a clump mass of white stuff. We pulled out some with a glass rod, and it looked like a tiny, clear piece of string. It did not look like the double helix shape that we see in most textbooks, because in reality, the individual strands are way too small to see with most microscopes, let alone the naked eye. The final step of this lab was to test the pH of DNA. DNA, or deoxyribonucleic acid, is of course an acid. This would have been proved by litmus paper turning red when the DNA was introduced to it, but we were unable to remove all of the solution from the DNA before testing. The basic pH of baking soda from the extraction solution messed up our results, turning the litmus paper blue instead of red, indicating a base instead of an acid.
Wow!